This is your instructor Kwame Shen. So for this lecture, which is part 1 of our lecture on microbial metabolism, we will look at some of the chemical reactions that take place in microbes, the energies that they're able to harness, the fact that some of them are able to use carbon dioxide, sulfur, and iron, the different metabolic pathways. We'll also gain an appreciation of the fact that most of these chemical reactions that take place that in microbes and that's help them to generate energy or build body parts are reactions that also occur in eukaryotes, including humans. So this is some, but not all, of the metabolic pathways. And our understanding of this in terms of the microorganisms will help us to appreciate how helpful they are to us, as well as how we can control them in instances where they cause diseases. So broadly speaking, we will look at these outcomes for this lecture. Enzymes, how they speed up reactions, what they do to activation energy, the different reactions that they catalyze, how enzymes are named, co-enzymes, enzyme environment in terms of turnover, the different types of inhibition that we do have, metabolism, what the energy carriers are and how they are generated, the different pathways in terms of glycolysis and fermentation and cellular respiration, as well as the alternative pathways in addition to glycolysis that some microorganisms carry out. And then we'll also look at anabolism, the processes that lead to the building of molecules in microorganisms and in eukaryotes as well. So metabolism as a whole is a description that we give to all the chemical reactions that take place and thus make it possible for living organisms to thrive, to survive and thrive. And so when you put all these reactions together, we call it metabolism. It is through these processes that food is broken down to release energy. It is through these processes that the energy that is generated from breaking down food is also used to build up the different components of the organism. Now, of course, in the process, waste is generated, and that waste is then released or eliminated from the organism. So nutrients play an essential role in metabolism. They serve as a source of energy, and then they also serve as the building blocks in terms of the different parts or components of the organism that need to be built together. We call it anabolism, a building together of molecules. So it will involve the breakdown of food to release energy that is stored in the food, and the bringing together of micromolecules to form macromolecules like proteins and carbohydrates that are needed for life. So for instance, if we take this banana, Lego banana here that we have, if it's broken down, it will broken down all the way to glucose, and in the process energy is used, but energy is also released, and more energy is released than is used. And this energy that is then released will then be used to build other components that the cells of these microorganisms need. And enzymes play a crucial role in chemical reactions. So these are biological molecules that are found within the cells of living organisms. what they do is that if we look at the theory of collision, which explains how reactions happen, that for reactions to happen, the molecules that are always in constant movement, which has to come together to form the reaction, need to have enough energy, and they have to have the correct orientation in terms of how two or more molecules are structured. So, for instance, we don't have any reaction taking place in this instance. However, because in this instance this molecule fits directly into this molecule here, and they have the right amount of energy, we have a product being formed. So this is a new product, and that's a new product. So a reaction has taken place. So there is constant collision taking place. You need the right amount of energy and the right orientation. Now, in terms of the energy, there is a minimum amount of energy that is needed for a reaction to occur. And that minimum amount of energy that is needed for reactions to occur and products formed is known as the activation energy. That's the activation energy of the reaction. So you should have sufficient collisions going on, but then the molecules or the reactants should have enough energy. And the minimum is the activation energy. What enzymes do is that when enzymes are present in a reaction or in any system, in this case, microbial organisms, what they do is that they reduce the amount of energy needed. The minimum amount of energy needed for the reaction to take place, which is the activation energy, is lowered. When the activation energy is lowered, it speeds up the rate of the reaction, so the reactions happen faster. Less energy is needed, so there's more collision that results in formation of products. What we should note, however, is that in the process, the enzymes are not permanently changed. They are never used up. They are recycled and used over and over and over again. So this is a typical cycle of what the enzyme goes through. So you have an enzyme. These are the substrates. When the substrate and the enzyme combine, it forms what we call the enzyme substrate complex. So you have the enzyme substrate complex here. When the enzyme substrate complex is formed, you end up forming a product. So you see the color changes indicating the changes that is taking place. You have a product here. The product is then released from the enzyme, but the enzyme remains the same. And then it enters the system again, picks up new substrates, and converts them or helps in the conversion of these substrates into products. And so this goes on and on and on. Most enzymes are protein in nature. Note that I did not say all enzymes, but most enzymes, because there is an exception. Now, enzymes govern or regulate reactions that involves the copying of genetic material or genetic information, maybe DNA, and then transmitting this information into a messenger RNA, and then using that information to form protein. We'll talk more about this when we talk about the genetics of in microbes. Protein has to be folded in the right way for it to be able to be functional. Remember, we talked about primary, secondary, and tertiary structures of protein. So if you look at the tertiary structure of protein, which is a 3D structure, that's how proteins are generally. For in this instance, we have an unfolded lysozyme. So it's an enzyme that is involved in the breaking down of molecule. You find this enzyme in the organelle lysosome. Now, so this lytic enzyme, for it to function, it needs the aspartic acid component of the amino acid, the tryptophan amino acid, the alanine amino acid and a um, glutamic acid amino acid coming together. Now, when all of them come together at the same time in the same area like this, then you have the active site formed. When it's not folded, look how dispersed they are. They're too far away from each other for them to form an active site, and thus this protein will not be functional unless it's folded correctly in this manner. So no shape, no reaction. If their shape is destroyed, they are not as functional as they should be. Enzymes then will have an active site, like the example that I just gave in the unfolded and folded enzyme. They are very specific in terms of the molecules that bind to them. Now, molecules that's attached to enzymes and therefore are affected by these enzymes at their active sites are known as substrates. So if we look at this example here, these are our enzymes. These are the active sites, and you see that they have different active sites. So a substrate of this shape cannot be catalyzed by this enzyme in any reaction because the active sites are different. So these active sites have to fit into the substrate for the enzyme to be able to catalyze that reaction. So enzymes are very specific. So you have a substrate, you have an enzyme, you have a substrate, it attaches to the active site. It forms an enzyme substrate complex, and then a product is produced in the process. Okay. How enzymes are named gives us a lot of information about what substrates they catalyze, what type of reaction it is that they catalyze, and you have the A's that is attached telling you it is an enzyme. So for instance, if we take sucrase is an enzyme. The name tells us that the substrate that it acts on is sucrose. The reaction that it's involved in is the breakdown of sucrose into glucose and galactose. And the A's tells us it is an enzyme. Similarly, if we look at DNA polymerase, the substrate is DNA. The polymerase, the polymer part of it tells you that it brings DNA together. It brings nucleotides together to form this DNA. Okay, so it forms a polymer, and the A's tells you that it is an enzyme. The same way with DNA ligase. So how an enzyme is named gives us a lot of information about what substrate it acts on, whatever reaction it is involved in as well. Some enzymes would need a secondary molecule in order for it to work. These secondary components are non-protein in nature. Note that most enzymes are protein in nature, with very few exceptions. So you would need both the enzyme and the non-protein component for it to work. Now, if the non-protein component is inorganic in nature, it is called a cofactor. So inorganic non-protein components that enzymes will need in order to catalyze reactions are known as cofactors. And these are some examples. So they are mineral in nature, they are inorganic. Zinc, manganese, and iron are some common examples. So in such an instance, you have an enzyme, you need a cofactor to be here, and for the enzyme to fit on the new shape that is formed when the cofactor attaches in order for the reaction to proceed. This is where the second non-protein component is organic in nature. So If they are inorganic, they are cofactors. But if they are organic, then it's called a coenzyme. Coenzymes are mainly made from vitamins and nucleotides. This is something that you should note as well, that when you only have the protein component of the enzyme, it is inactive, and therefore it is referred to as an apoenzyme. However, when the coenzyme attaches to the apoenzyme, it becomes a holoenzyme. A holoenzyme means that it's now activated. It's created an active site where a substrate can then bind to, and then the reaction can proceed. There are some important coenzymes that exist in living organisms. So a few of them are these, which you'll come across in this lecture on microbial metabolism. So we have nicotinamide adenine dinucleotide, NAD+. You have nicotinamide adenine dinucleotide phosphates, and these are important protein carriers. What you will note here in the structure is that everything is the same except when it comes to the nitrogenous base, where you have 2 hydrogen here, which makes it reduced. So this is reduced nicotinamide adenine dinucleotide, and then you have only 1 hydrogen attached here. Then you also have coenzyme A, which is also very important in the process of cellular respiration. Enzyme actions are regulated by environmental conditions and some other factors as well. So we'll look at some of these factors that regulate enzyme activities. Generally, enzymes are able to really ramp up the rate of reactions significantly, that a reaction can actually be sped up to about 10 billion times than it would have proceeded without an enzyme. So enzymes will react in ways that significantly improves the rate of reaction, and therefore makes it possible for living organisms to maintain their lives and continue to pass on genes to the next generations. And so when we're talking about how efficient enzymes are, we talk about them in terms of turnover number, that how many substrates is the enzyme able to process within a second. So if we take DNA polymerase, 15 molecules are processed within a second, so 15 nucleotides are processed in a second. If we look at lactic dehydrogenase, which is an enzyme that converts lactates to pyruvate, and it's a reverse reaction, so it goes in both the forward and reverse direction. It will process 1,000 lactate molecules per second, and so the difference is very, very phenomenal. It's great. We look at 2 instances where you have low enzymes. So you have the same, you have 5 substrates, you only have 2 enzymes that are available in terms of molecules. The rate of reaction, because you don't have the right concentration, will be slower. However, if you increase the concentration of the substrates, it gets to a point where even if you increase it more, it doesn't really make much of a difference, because the rate at which the enzyme processes these molecules would be constant. But you have more molecules for enough interaction to take place with the active sites, then the rate of reaction would definitely increase. There are some environmental conditions that also affect how enzymes perform. We have temperature, we have pH, and the concentration of the substrates. So if you look at temperature, we talk about temperature mainly in 3 terms. We have optimum temperature. That's the highest temperature or the temperature at which the enzymes perform best. So the maximum output is at that temperature. And for most microorganisms, the enzymes that regulate their reactions or catalyze their reactions would function best between 35 and 40 degrees Celsius. You would notice that the human body optimum temperature is around 37 thereabout degrees Celsius. So these organisms are generally will be able to live on living organisms. Then for every enzyme, you have the lowest temperature beyond which the enzyme will not be able to catalyze a reaction. And the same can be said for the highest temperature above which the enzyme will not be able to catalyze a reaction. Now, for very high temperatures, it is very likely that these proteins, which are mostly these enzymes, which are mostly protein in nature, would be destroyed permanently and therefore cannot perform their reaction. So graphically, if we look at the rate of enzyme activity and the temperature, as you continue to increase the temperature, if the temperature is below a certain value, the enzyme will be inactive. But at a certain value in terms of temperature, the enzyme is active and therefore can catalyze reactions until it gets to the point where it produces the most product, which is the optimum temperature. If it goes beyond optimum temperature, it gets to a point where the enzyme will then be destroyed and therefore cannot catalyze reactions again. The destruction of enzymes, either by environmental conditions, mostly by temperature, is referred to as denaturation. It destroys the folding, and once the folding is destroyed, the enzyme can no more function. Then there is pH. Most enzymes will function best at a pH of 7, which is neutral. There are instances where you have too much hydrogen ions that are present and therefore clog up the active site of the enzyme, and therefore the substrate cannot bind. And the same can be said of conditions where it's too alkaline, there are too much OH minus ions, which same way clog up the active site, and therefore the substrate is not able to bind and the reactions do not proceed. But in all this, there are exceptions. So if you take stomach enzymes, you have a very high concentration of hydrogen ions there. You have HCl, so we have a pH of 1 to 2, but that is the environment that those enzymes can actually perform best. Then there is substrate concentration. The substrate is the substance that the enzymes act on. Now, if we have a certain concentration of substrates compared to the enzyme, for every reaction, it gets to a point where even if you should add more substrate to a certain concentration of the enzyme, the reaction rates cannot be sped up. So that point is known as the point of saturation. At the point of saturation, you have the right amount of substrates for that concentration of enzyme that is available or that is present. And there is enough interaction going on between the active site of the enzymes and the substrate. So even if you should add more substrate to it, you cannot increase the rate of reaction because there isn't more enzymes available. The only way you can speed up is that you would increase the concentration of the enzyme. So for a specific concentration of enzyme, there is a point beyond which even if you should add more substrate to it, to that system, the rate of reaction will not be sped up. And that is the point of saturation. So let's do a learning check. If you have sucrase, which has an optimum temperature of 37 degrees and optimum pH of 6.2, and then you are to determine the rate of reaction of of sucrase. If you increase the concentration of sucrase, so generally at a specific concentration, if you increase the concentration of sucrase, there should be an increase in the rate of reaction. Okay, you've increased the substrate concentration because substrates you increase the concentration of the enzyme, and so once you increase the concentration of the enzyme, the rate of reaction would also be faster. And then if you change the pH to 4, so it's at 6.2, which is closer to neutral, and then you bring it to 4, which is more acidic, relatively speaking, what happens to the rate of reaction? Because you've changed the pH, what will happen is that there will be a decrease in the rate of reaction. What about when you increase the temperature from 37 degrees Celsius to 70 degrees Celsius? If you increase the temperature to 70 degrees Celsius, most likely you will denature the protein in the enzyme, the sucrase enzyme, and therefore the rate of reaction will decrease. So it's important that you understand what's going on here. If you increase substrate concentration, or if you increase enzyme concentration, if you increase pH, if you increase temperature, or if you decrease pH or you decrease temperature, what happens to the rate of reaction? Okay. Enzymes are regulated, and they are regulated in order to control the amount of energy that are released and the resources that are used in building up different components of the organism for it to maintain life. For enzymes to be regulated, there should be some mechanism in place that makes that possible. And so we'll look at 3 of these mechanisms. We have what we call competitive inhibition, we have allosteric inhibition, and then we have allosteric activation. Now, if we say some molecule is an inhibitor, it means that it prevents, in this context, it prevents the enzyme from doing its work. So if we say competitive inhibition, we are talking about an inhibitor, another molecule that competes with the substrates and thus prevents the substrate from attaching to the active site of the enzyme. There are 2 types of these competitive inhibitors. You have the reversible and the irreversible. So typically, for a regular reaction without any inhibitors, you have an enzyme, you have a substrate, that's the active site, it attaches to it, it forms an enzyme substrate complex, and then a product is produced as a result. However, if you have a competitor that is competing for the active site with the substrate, that's typically what will happen, is that you have an enzyme, you have an active site, and then you have an inhibitor. The inhibitor will then attach to the active site. You have a complex that is formed that is not an enzyme substrate complex, but an enzyme inhibitor complex, and therefore the substrate is not able to bind to the active site, no products are produced, and so the reaction does not proceed. So for the reversible types of competitive inhibition, what happens is that whenever there is an increase in the substrate concentration, so you have the competitor, you have the substrates. If we should ramp up the concentration of the substrate, what happens is that you have more substrate competing with the inhibitor, and therefore the probability of the reaction taking place and proceeding increases. So this is an example of a reversible competitive inhibition reaction. Now, for the irreversible, even if we should increase the concentration of the substrates, the inhibitor is not overcome, and therefore it can continue, and thus continue to inhibit the reaction from proceeding. There are important practical uses of this phenomenon. So, for instance, we have PABA, which is the full meaning is para-aminobenzoic acid, and para-aminobenzoic acid is used by several bacteria as a nutrient, and 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 because it's used as a nutrient, if there is a way that we can create another molecule that will compete with the active site of the enzyme that catalyzes this reaction, then we'll deny the microorganisms the nutrients that they need. The good aspect of this particular treatment is that human cells do not use PABA, PABA, and because human cells do not use para-aminobenzoic acid to make folic acid, which is used in making nucleotides and nucleic acids, if we use these drugs on these microorganisms, it does not affect humans. So that is an example of a magic bullet that Ehrlich was talking about. Now, what happens is that you have an enzyme, you have the active site, and you have PABA or PABA being able to attach to the active site, and then the reaction proceeds. However, if you have sulfur drugs, which generally we call the sulfanilamides. Now, these sulfanilamides would then serve as competitive inhibitors and prevent PABA from attaching to the active site. And therefore, the organism is denied folic acid, which it will need to create nucleotides and nucleic acids, and therefore they are killed by these drugs. These drugs were used heavily during the Second World War to treat wounds of soldiers and be able to prevent the infections from happening. Then you have penicillin. Penicillin is an example of irreversible competitive inhibitor. And what happens is that it covalently it's able to bind to the sites where the synthesis of the peptidoglycan is taking place, and therefore prevents the formation of the cell wall. If bacterial cell wall doesn't have the integrity that it needs, then it's very easy for it to break down. So for instance, if we look at a scenario like this, you have penicillin that will bind to the site and therefore prevents new molecules to be added in synthesizing the cell wall of the bacteria. So that's an example practically that exists. Other types of inhibition that we have, we call this allosteric inhibition. And for allosteric inhibition, the competitor or the inhibitor does not bind to the active site. So that's one major difference. The inhibitor binds to what we call the allosteric site. So this is the enzyme, that's the active site. The inhibitor binds to this site, known as the allosteric site. However, once it binds to the allosteric site, there is a change in the shape of the active site. So there is a conformational change, and because there is a conformational change in the shape of the active site, the substrate is not able to bind. So the active site changes, the substrate is not able to bind, and the reaction does not proceed. And so these are the types of inhibition that we've looked at. We've looked at normal. You have a substrate, you have an active site, it binds to the enzyme, and the reaction proceeds. And then we've also looked at instances where you have competition. It could be either a reversible or irreversible competitive inhibition. Now, in these instances, you have another molecule that competes with the substrate for the active site. Then you have non-competitive inhibition, and that's an example of allosteric inhibition, where you have an allosteric site that the inhibitor binds to, and therefore changes the shape of the active site, and therefore the reaction does not proceed. An example of allosteric activation. So we're talking about allosteric inhibition. There is also allosteric activation, where you have a molecule that attaches to the enzyme, and once it attaches to enzyme, it also changes the active site, but rather makes it more accessible to the substrates. So this is an inactive enzyme. You have a molecule that attaches to the allosteric site, and once it attaches it then activates it. So now you have a hollow enzyme, and then the substrate is able to fit, and then the reaction proceeds. So there is allosteric activation as well. And an example of that is hemoglobin. Hemoglobin is an allosteric protein. Once the binding of oxygen to one hemoglobin subunit occurs, it induces induces the other molecule, makes it easier. There are changes that take place, makes it easier for the other molecules, the subunits, to pick up oxygen, and more easier as more oxygen attaches. So if you see the changes that is taking place in terms of the shades of the color, in this instance there is no oxygen attached. However, once you have one molecule attached to hemoglobin, it starts changing the conformation of the other molecules in the group. And so the second molecule attached, the hue continues to change, another molecule is attached. So this is an example of allosteric activation. These results in production of end products. But the production of end products, it gets to a point where there is enough product produced that the organism should be able to regulate the amount of products that is being produced. And so as part of its regulation, certain molecules, once they're produced, then serve as an inhibition. And an example is what we call the feedback inhibition or the end product inhibition. It's also an example of allosteric inhibition. However, in this instance, it is the product that is produced, the final product that is produced, that becomes the inhibitor. It then binds the allosteric site and prevents more of its kind to be produced. So this is how it goes. You have an enzyme, you have a substrate that attaches to the active site, you have several intermediates that are produced, but then you have the end products that is produced here. Once you have the end products that is produced, the end products then attaches to an allosteric site on the original enzyme and changes the active site. So the active site is changed from this angular shape to more of a rectangular shape here, and that makes it impossible for this substrate to attach, and then the reaction is stopped. Practically, this happens in E. coli. E. coli synthesizes isoleucine, which it needs for cell growth. But as isoleucine is controlled by feedback inhibition or end product inhibition. So you have the enzyme, that's the active site. You have threonine, which is a substrate that attaches to it. You have a complex, and there are several intermediates and several enzymes involved. But then you end up with the end product, which is isoleucine. Once isoleucine is produced, it attaches to the allosteric site, changes the conformation of the active site. Now, once the active site's conformation is changed, what happens then is that the substrate is no longer able to bind, and therefore the reaction ceases. Up until 1982, we believe that all enzymes were proteins. However, in the 1980s, Altman and Church identified some enzymes that are not protein in nature. So these are enzymes that are made up of RNAs, ribonucleic acids, and they're able to catalyze reactions as well. They are used in RNA splicing, and so we'll talk about this during the genetic engineering lecture. This brings us to the end of part 1 of microbial metabolism.